Everybody to the December 2022 edition of the BC Associate Speaker Series. This is our last one of 2022. The year's flown by, as I'm sure we can all relate to. So we have, and this is the last talk for December 2022, but we have 4 more scheduled, 5 more scheduled for 2023 in the springtime, winter, spring, and we'll be starting up again in the fall from there. And I apologize that I have a cold, so my voice is not very good. And I want to take a moment to recognize that many Indigenous nations have long-standing relationships with the territories upon which York University campus is located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Tikaranto has been caretaken by the Anishinaabeg Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis peoples. It is now home to many Indigenous peoples, and we acknowledge the current treaty holders, Mississaugas of the Credit First Nation, and this territory is subject to the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. And especially as this is a webinar, people are joining in from all across Canada and possibly beyond. So I encourage you to consider where you are located currently or where your research has occurred or where your specimens are from, and think about the people who have caretaken that land. If you aren't familiar with the people who are traditionally or currently on that land, visit www.native-land.ca to have a really good interactive map that you can then browse to see who are the caretakers, past and present for your region. And with that, I want to introduce our speaker today. Just let me get her bio up. Katherine is a second-year postdoctoral fellow in the Rehan Lab at York University. She is interested in using bioinformatics to capture genetic information of the metagenome and metatranscriptome of crucial species, such as wild bees, to gain insights on the important interactions and relationships between microbiota and host. Previous to this, she completed her PhD at the University of Toronto, where she analyzed selection patterns on various genes on neotropical fish. She completed her master's at the University of Windsor, where she was first introduced into bioinformatics, analyzing environmental DNA of water samples from the Great Lakes to detect at-risk, common, and invasive freshwater fish. And with that, I'll turn it over to you, Katherine.
Okay, all right. Thank you, Victoria, for the introduction. Okay, so I'm going to talk about the work that I've been doing my postdoc is at Rehan Lab at York University. My talk will address 2 main projects I've worked on and am working on. The first part will be metagenomics and some popgen of the small carpenter bee, Ceratina calcarata, which I'm sure we're all familiar with, seen on the picture here. And the second project addresses the developmental transcriptome and metatranscriptome of the same bee. Let's get my laser pointer out. Okay. All right, so the first project, again, focuses on metagenomics and how it's impacted by urbanization. So bees play a crucial role in their environment. They are vital to sustaining healthy, diverse ecosystems and provide the important service of pollination of our essential crops. They also act as indicator species, so if their populations are declining, likely there's something wrong occurring in the environment. Anthropogenic practices such as agriculture, urbanization, and human-induced climate change are all a cause for global declines in pollinator populations. But what's more concerning is this expectation by year 2050, 2/3 of the global human population will reside in cities. So this means that more natural land will be transformed into cities, leaving unmanaged wild bees vulnerable. To help combat pollinator population declines, especially in bees, we need to fully understand their ecology and what specifically contributes to healthy bee populations. This is a multifaceted approach and includes different fields of research, which many in our lab are undertaking right now, and includes really cool research like phylogenomics. So it helps us understand their evolutionary history and explain why bees are the way they are today. Utilizing transcriptomics to study gene expression patterns and how bee genes are regulated or altered with the environment. There's research on bee ecology, like their behavior, which helps us understand how bees act with each other or, again, under different conditions. There's population genetics, which informs us of the distribution of populations and their structure. And finally, taking a metagenomics or metatranscriptomics approach, which is the focus of my research, where the aim is to determine who else is there or who else is actively interacting with bees and what might those interactions mean for the bee. And here we usually analyze or talk about the bee microbiome and metagenome. So microbiomes and metagenomes, they consist of the community of microorganisms found in an environment. And in bee research, a lot of the time, this is focused on the bee microbiome. So this is the community of microorganisms found within the bee, which typically includes bacteria, fungi, and viruses. And then the metagenome includes these taxa, but it also includes associations with all other organisms that the bee might come into contact with externally. And these can include the plants that they forage on, protists or mites and arachnids, or nematodes or other things that could be potentially found within their nests or that bees are interacting with. And research has shown that bee microbiomes and metagenomes play an important role in bee health. Bee microbiomes contain microfauna that are crucial in bee development, their nutrition, digestion, immunity, as well as cognitive ability. And then bee metagenomes provide essential information on other environmental relationships, for example, important plant and pollinator networks. So what plants are bees visiting the most? And in either case, one of the major revelations with this type of research is detecting any harmful taxa like pathogens and parasites that might be associating with bees. So for the first project, I explored PopGen and the metagenome of the small carpenter bee. So the genus Ceratina is globally distributed, but Ceratina calcarata is found in North America across rural and urban locations. It is a subsocial native species and often classified as solitary in that it typically is a mother and her offspring in a nest, so it's not a massive colony. The mother will excavate a linear tunnel in the pith of dead stems, most popularly in raspberry or sumac, laying one egg at a time on a pollen mass provision that she's foraged for beforehand, before closing off the cell using pith shavings. And then offspring will develop while mothers remain, guarding and cleaning the nest and reconstructing cells as needed. And because they're classified as solitary, their microbiomes are mainly obtained from the environment and their diet, unlike in highly social bees where their microbiomes are heavily influenced by social interactions between colony members. And just to add, these species, they're wild and they're unmanaged, but they are still just as important to monitor and observe as compared to the more well-known managed honeybees and bumblebees. So out of the approximately 25,000 globally identified bees, close to 20,000 are wild, so they're unmanaged, and about 75% of bees are solitary. And while honeybees and bumblebees are excellent pollinators because they're generalists, can pollinate a large range of plants and crops, for certain plants and crops, native and solitary species are more efficient at pollinating them, once again highlighting the importance of examining the health of bee populations of all types. And so with that said, we need to further study how wild bees respond to increased urbanization, which not only alters green spaces, but it also creates urban microclimates, the most prominent being the urban heat island effect, where temperatures are unnaturally warmer in cities. This is not optimal for most bees or even for most species. And so to help us obtain a better understanding of how they're responding to these changes, integrating approaches like population genomics with metagenomics, we can use this combined approach to, for example, detect dispersal corridors in urban environments. We can assess bee microbiomes and metagenomes, and then we can also use metagenomic info to detect those important interactions like any potential pathogens and parasites. This in turn will give us a clear understanding of bee health in along an urbanization gradient, identify favorable land conditions for bees, identify notable co-occurring taxa, for example, important plants that urban bees need, and of course, disease monitoring for those harmful co-occurring species. So this brings me to the aim of my first project, which is to understand how the urbanization gradient influences wild bee genetics and ecological interactions. I have three aims, and the first is to estimate genetic diversity and population structure and assess how differences in landscape features affect gene flow across the urban gradient. Here we hypothesize that population structure within the city is driven by landscape features, and that areas dominated by vegetation will facilitate Ceratina calcarata dispersal. The next aim is to determine how differences in landscape influence the metagenome and microbiome of the bee. And here, we expect differences in taxa diversity across the urban gradient. And finally, we want to identify potentially parasitic or pathogenic taxa to urban bees. Here, we hypothesize urban sites will harbor more potential pathogens relative to rural sites. Okay, moving on to my methods. So Evan, who's one of the co-authors for this first project, had gone sampling for female Ceratina calcarata during the provisioning season from August to November in 2019 and 2020. Here in this picture, you can see him trekking through some shrubs, some small trees and grasses. And this picture really captures the ideal habitat for small carpenter bees because there's a lot of nesting material for them to burrow in. In total, 180 females were sampled from 66 sites across the Greater Toronto Area, or GTA for short, which were previously chosen to cover a broad landscape range. And so this includes a range of developmental percent, where 1 would indicate very low levels of development, 5 would be very high levels of development. And in general, what we see here is around in the center toward the waterfront, development ranges from typically moderate to very high, and then further away, it decreases. Conversely, we see the opposite pattern for green space coverage, which makes sense. Typically, where development is very high, we see that green space coverage is low, and vice versa. Our sites also range in annual precipitation. So further away, again, moving away from where development is moderate to high, precipitation increases, and precipitation is low, where development is typically high and green space is low. And the sites also range in temperatures. So towards the waterfront here, where development is high, temperatures are also very high. And then moving further away, temperature decreases. Next, the sampled females were thoroughly cleaned off with ethanol, and DNA extraction was done on the whole bee specimen for whole genome sequencing using Illumina NovaSeq. Reads were then checked for quality, trimmed, and mapped against the Ceratina calcarata reference genome. Next, we used a program called Metaspades, which obtains context for all of the unmapped reads, which was used for metagenomic analysis. And then all the mapped reads were used for PopGen analysis. And this was done by Farida, another co-author in the study. And just focusing a little bit more on the unmapped context, because that's sort of the part of the project I focused on more. They were then blasted for taxonomy classification and then filtered to focus on 7 domains or 7 groups of interest of taxa that are known to primarily interact with bees or associating with bees. This includes bacteria, fungi, nematodes, viruses, arachnids, protists, and plants. And finally, we ran various analyses to identify any interesting patterns with our metagenomic and PopGen data with respect to varying urbanization. So now I'll dive into the results and discussion at the same time. As a reminder, our first objective was to understand population dynamics of our bee along the urban gradient. And surprisingly, what we found was that across our urban sample region, it was found to be one large panmictic population with relatively high levels of inbreeding. This disagreed with our earlier hypothesis that urbanization would promote population fragmentation, especially in this small solitary bee. So next, we also analyzed resistance scenarios, and this asks how do different landscape features, for example, croplands, grasses, bare ground, urban land, affect bee dispersal? In this case, our results agreed with our prediction that vegetation-rich land types, so trees, shrubs, scrubs, grasses, and crops, would facilitate bee dispersal. When we visualized our resistance landscape values in the second plot here, in the highly urban site, so again, in the center of our sample region, we see that the dispersal corridors become much more limited, depicted by these red pathways, as opposed to these areas further out. These limited pathways are likely urban parks, small green spaces, residential gardens, and it really emphasizes that although scarce, these vegetation-rich landscapes are vital for urban bee movement because we do see this one large population. Next, we also looked at alpha diversity in terms of richness along the urban gradient to determine how differences in landscape influence the metagenome of the bee. So in this case, we did see that overall alpha diversity across the majority of the domains were highest in those densely urban sites. So where there is high temperature, high development, but low precipitation and low green space. This agreed with our expectation that we would observe differences in taxa diversity across the urban landscape. But even more so, we see that the largest group, bacteria, which was actually the largest group in terms of abundance in our metagenomic data, drove most of the diversity pattern we observed. And in fact, another recent paper from our lab by Cindy and Sandra in 2022 also looked at bacteria and fungi abundances in Ceratina calcarata from similar sites, and also found that bacterial richness was greater in highly developed sites compared to low development sites. So there is a clear pattern here where urbanization is impacting, at least with more clarity, bacterial microbial abundance and richness in urban bee microbiomes. So next, in this figure, we generated a heat map with the average number of genre for each of the domains for each of our environmental features. This figure shows us that the highest average values for genre are clustering around these environmental features that are really representative of urbanized sites. So where there is high temperature, high development, low green space, and low to moderate precipitation. And then our final objective in this project was to identify potential pathogens to urban bees. And this is also where we combine population level with landscape and metagenomic level data to assess whether urban sites harbor more pathogens. Here we search for instances of pathogens known to be harmful to bees and to plants, because plants can also harbor many pathogens and parasites affecting them, which in turn can indirectly be detrimental to bees. So in this case, we found that urbanization does have an increased risk of pathogen exposure to wild urban bees. Similar to the previous heat map, this heat map highlights that the high average numbers of pathogens and parasitic genera for plants and bees are highest in landscape features, characteristic of highly urban sites. So once again, that's moderate to low to moderate precipitation, very high temperature, very low green space, and very high development. At the same time, we ran a correlation network analysis on pathogen diversity. We compared that against PopGen and landscape parameters, which included relatedness, environmental distance, and resistance. Here we found that the only significantly and positively correlated variables were between pathogen diversity and resistance, emphasizing again that there is greater pathogen load in those densely urban areas, where resistance to dispersal is also the highest because it's much more fragmented there. This supports our hypothesis that urban sites would harbor more pathogens relative to rural sites. And so just to conclude, overall, we found that the carpenter bee has high levels of inbreeding, but remains to be one large panmictic population, likely due to the very important but scarce vegetation-rich corridors facilitating its dispersal. The reduced genetic diversity is actually something that's been found in other rural and urban bees, and it would be something to explore in future studies to assess how reduced population differentiation is impacting urban bee fitness. So while low genetic diversity may be common in bees, it actually can be quite detrimental in urban bees, simply because gene flow is already more limited due to it being a fragmented landscape, leaving these populations more susceptible to issues like bottlenecks. Next, we found that taxa diversity across the domains were generally higher in urban areas. And in particular, this was true for microbial, so bacterial diversity. This may be problematic as many bacteria can be harmful to bees, and urban settings can increase exposure to that type of bacteria. And indeed, we found that densely urban areas, which have increased resistance to dispersal, also had increased pathogen diversity and load. The silver lining here was that in our current study, the pathogens that we did detect and use They were present at very low abundances, so this may offer an early opportunity for early pathogen and disease biomonitoring. And overall, what we learned is that integrating PopGen work with metagenomics is an important tool in wildlife conservation. It helps us determine population structure and important interactions, especially pathogen detection, especially early pathogen detection. And this type of approach is very useful for future research and management projects, as it can help us establish predictive models for suitable habitat and really prioritize which areas or those which type of corridors, so those vegetation-rich corridors, should be the focus of restoration efforts. So right now, I'll switch gears to my second project, which I'm currently working on. This time, the focus is more biology-driven, and here we look at the developmental transcriptome and metatranscriptome of Ceratina calcarata, and how it may be influenced by the presence and absence of mothers. So just really quickly, gene expression in simple terms, it is the process in which information in DNA, so our genes, are activated in a cell, allowing the cell to produce proteins and perform specific cellular functions. It is the process by which genotype, our genetic blueprint, gives rise to phenotype, our observable traits. And then changes in the environment, so for example, rich or poor nutrition, changes in temperature, presence or absence of parents, can impact gene expression, and this in turn will give rise to different phenotypes, also known as developmental plasticity. In other words, the same genotype can encode for multiple phenotypes, but it's depending on the conditions of the environment. Now, focusing on parental care, it plays a significant role in how individuals develop in species that have evolved to depend on some form of parental care. The neurobiological and behavioral effects of parental care have been widely researched, and various forms exist. So, for example, maternal care is the dominant form of parental care in most insects, for example, in solitary bees and in earwigs, as shown here, where mothers will perform nest building, food provisioning, and take care of the young. In humans and birds, biparental care is dominant, so both the father and mother take some productive role in raising their young and protecting them. And then there are cool forms of parental care that have evolved, for example, in mouth-brooding cichlids, where paternal care, so fathers take care of the young, they will actually hold their larval offspring in their mouth as a form of predator protection. And so, as I just mentioned, in solitary bees, maternal care is prevalent. Mothers perform nest building, pollen foraging, and they also display nest loyalty. So they will continue to raise offspring in the same nest until the offspring reach adulthood and fly off to form their own nest. Additionally, deprivation of maternal care has been shown across several animals to cause long-term behavioral abnormalities. And in fact, in a 2018 study, they observed adult Ceratina calcarata that were raised either with or without mothers, so care or no care treatments. Adults reared without mothers showed differential gene expression compared to those with mothers. There were also subtle changes in in DNA methylation, with slight increase in DNA methylation in cared individuals. And overall, what they found was that adult Ceratina raised without mothers on average had increased aggression and increased avoidance. This is thought to occur as a response to not having any adult kin in the nest. And actually, this type of behavior would help to improve their overall survivability due to the lack of mothers. And now just to touch on some points about microbiomes. Microbiomes are also known to change through development of the host. This has been researched widely in a variety of species, especially in humans. So in humans, it's well known that our gut microbiomes clearly change from when we're newborns to toddlers to adults and elders. And it's a lot of it has to do with the changes we go through in our environment and in our diet. With that said, a paper from our lab explored how the microbiome changes in the small carpenter bee through development. And what they found was that with bacterial richness, it tended to decrease as brood developed into adults. And interestingly, fungal richness was fairly similar across stages. However, fungal composition did change across developmental stage, where some taxa that were found during the larval stage were not found in later stages. This finding shows that overall diversity and composition of the Ceratina calcarata microbiome does change with development. And so, with that said, while maternal care effects were explored in Ceratina calcarata at the adult stage and the observed gene expression patterns and its microbiome was explored through development, we do not know what are the changes in gene expression patterns in the species across development with respect to maternal care, nor do we know how maternal care specifically impacts the developmental microbiome. So this brings me to the aim of the second project, where the goal is to understand how maternal care impacts gene expression and the microbiome across development of Ceratina calcarata. There's also 3 aims. First is to characterize the developmental transcriptome and test how maternal care impacts gene expression patterns. Here we expect a differential gene expression between stages and care groups, and we also expect developmental genes to be highly expressed in earlier stages. Next, we want to examine the changes in microbiome diversity and composition across development and between maternal care groups. Here we expect differences in microbial diversity between stages and groups. And we also expect greater abundance of bacteria and fungi in no care simply because mothers are not there to help keep the nests clean. And finally, we want to identify gene regulatory networks and co-expression with metatranscriptomic data across development. Here we predict dysregulation of gene expression and altered metagenomic microbial communities in the absence of maternal care. So now I'll talk about the methods. First, weekly nest collections were conducted by Miriam, another co-author on this paper or in this project. Nests consisted of sumac or raspberry stems, and then half the nests, eggs were taken out and reared in the lab in tubes, and these were the no-care treatments. And they were reared to the different developmental stages. And then the care group consisted of immediately flash freezing individuals taken from nests at different developmental stages, as they would have already been raised with the presence of mothers. Next, RNA extraction was done on 190 whole body samples. So we had 95 no care individuals and 95 care individuals across various developmental stages. And I'll just mention here, Ceratina calcarata has 19 developmental stages, but if you include the egg, it makes 20 stages. Next, extracted RNA reads were mapped to the Ceratina calcarata reference genome, and then all mapped reads were used for transcriptomic analysis, and all the unmapped reads were used for metatranscriptomic analysis. So similar to the first project, I again used metaspace to obtain context of all the unmapped RNA reads, which were then used for similar taxonomy classification. And then differential gene expression analysis, which was done by Miriam, was done to detect any differentially expressed genes, or DEGs for short, across the stages and care groups. Other analyses we did consisted of looking for clustering of certain groups of genes or taxa, or any overrepresentation of specific genes in taxa across the different stages and between the care groups. And finally, we also employed an integrative analysis approach to specifically assess how some genes in taxa, how do they correlate with each other between the care groups and stages. Okay, so moving on to results and discussions. For our transcriptomic results, a program was used to survey gene expression changes across development to obtain generalized linear models. And this essentially shows us how clusters of genes and the expression patterns, how do they peak through development? And so here we found 4 generalized clusters of time course gene expression, which ultimately summarized the 19 developmental stages into 4 overall developmental stages. So 1 is early larvae, 2 is late larvae, 3 is pupae, and 4 is callus. Next, a PCA plot showed that the first 2 principal components explain 66% of the variance in expression across stages. And we see that clustering is more pronounced for gene expression, is more pronounced for development rather than just maternal care group. So next, I'll be going through some volcano scatter plots, which just show us visually the main differences in gene expression data for each of the stages and the magnitude of the differences when looking at no care versus care. And so just to quickly summarize what we're seeing, across all stages, we did see differential gene expression, but by far during early larvae, it had the most significant differentially expressed genes. So again, DEGs for short, there were about over 3,000 DEGs. Specifically, the absence of mothers resulted in more DEGs compared to those reared with mothers. And this included genes that were involved with immunity, digestion inhibition, and for instance, the genes that were upregulated when mothers were absent included some cool genes like trehalase and venom acid phosphatase. So trehalase is responsible for converting a storage carbohydrate, trehalose, to glucose, and this is used for carbohydrate and energy metabolism. This gene is also found upregulated in mice that experience oxidative stress. And then venom acid phosphatase is found in bee venom. It has a protective and immunitive properties. And in fact, it's one of the reasons, one of the many enzymes in bee venom, which can cause an allergic reaction in humans who are sensitive to bee stings. And so these results here indicate that early larvae without mothers are associating with these immune defensive and stress response genes, most likely because lack of mothers is instilling a stressed environment. Next, for late larvae, there are about 480 DEGs, and those reared with mothers had genes involved with homeostasis, whereas cared late larvae had some DEGs involved with pupal morphogenesis. An interesting gene that we found that was upregulated in no care was a potassium voltage gated channel protein, also known as SHAB for short. And this gene is very much involved with homeostasis and regulating neuron excitability. So it's an important gene in maintaining action potentials in cells and returning cells to their normal state. So the upregulation of this gene is likely a response to maternal deprivation as is trying to maintain homeostasis within stressed cells. Next, just going through pupae, it had more DEGs upregulated in no care treatments and included genes involved with insect immune responses. And then also we had looked at Go enrichment terms for the genes. And what was interesting is that in no care pupae, DEGs Go enrichment included terms such as negative regulation of immune response, whereas in cared, Go enrichment were a representative positive regulation of eye photoreceptor development and other developmental phases. And finally, callos also had more DEGs in no care and included an interesting gene called hexamerin, which is also part of the immune response in insects and actually is found to help fight bacterial infections in other insects. Another gene called CAMK2 was upregulated, which is linked to memory impairment as it was observed in fruit flies. And so this suggests that a lack of maternal care may be affecting memory ability in adult callos. And so with this, we can say that, yes, we were able to characterize the developmental transcriptome, and it is evident that maternal care does have some impact on gene expression patterns, especially in the early larval stages, with many more DEGs highlighted. But again, all stages do show some differences in DEGs between care groups. Our second objective was to examine the microbiome across development and between care groups. Here we find that, yes, the microbiome changes across development and between care groups, especially so during the early larval stages. First, across all stages, there was greater richness in the larval stages, and this drops drastically in the pupal to callo stages. And this is likely because at pupae, they defecate, and this process removes a lot of the gut microbiota so that their microbiome becomes much reduced until their callos picks up a little again. But clearly, the early larval stage, there's much greater taxa richness in individuals raised without mothers, depicted by the black bars here. When we break this down further into the individual groups, by far, fungi are driving most of this variation, where there's much greater fungal richness in no care early larvae. And then in late larvae, this switches to more richness in the care groups. And in fact, we see this increase in richness in cared individuals also in pupae and callo stages. We also see a similar pattern in bacteria as fungi, but their richness counts are not to the same level as fungi. In fact, fungi contributed to 85% of the microbiome composition. And then the second most abundant group was bacteria, which only contributed 8%. So fungi clearly have an important role in the wild bee microbiome, especially during the larval stages. Additionally, one of the top fungi to contribute to most of the variation observed in early larvae between care groups was a potentially pathogenic plant fungus called Aspergillus. There was a significant difference in abundance of this fungus between early larvae care groups, and it was much more abundant in no care early larvae. Aspergillus can be problematic. It's known in honeybees to cause stone brood disease, where it mummifies larvae. It isn't known to do this currently in solitary bees, but it is known to indirectly harm solitary bees because it causes pollen spoilage. And so our results show mothers are potentially actively keeping this and other fungi at lower abundances, especially during early larval development, which may indicate that this stage is most important for ensuring survivability of the young. The next slide here, I just want to show another analysis which we ran, which is random forest machine learning. And here we wanted to compare how well transcriptomic data or metatranscriptomic data can be used to predict developmental stage or care group of the offspring. Now, compared to gene expression data, the ability of metatranscriptomic data, abundance data to predict developmental stage was less accurate. In fact, gene expression data nearly reached 100% accuracy when we are withholding about 90% of the samples. So the x-axis here represents how much of the data was withheld or kept in order to train the algorithm, and then the remaining percent is the testing data. And then the accuracy of the model is on the y-axis here. So as it increases, the model is more accurate, it's better. However, when predicting maternal care groups, so the blue lines here, both data sets were somewhat similar. They were about 70%, around 80 to 90 withheld data. But I will point out that using microbiome data, predicting maternal samples based on maternal care group was slightly better than it trying to predict developmental stage. And this is likely because of the strong effect of mothers during the early larval developmental stage and how it's affecting fungal and bacterial abundance. Whereas gene expression is most affected by developmental stage. And again, this makes sense because there are clear differences in development as offspring go through the different stages. Mothers do have some impact, as we've seen, but not so much to the point that we experience no growth of individuals at any of the stages without maternal care. So gene expression is really good at predicting offspring based on their stage. And then I would say metatranscriptomic data is better, slightly better at predicting offspring based on care group, but overall gene expression data did perform better. And then just our final objective, it was to identify links between genes and microbiome data. So in this case, I had used an R package program called Mixomics, which uses a sparse partial least squared regression analysis. And simply what this does is it integrates two data sets that are measured on the same individuals to detect highly correlated positive or negative variables. In our case, because we had so many DEGs to work with and hundreds of genera to work with, when we used the whole data sets, it resulted in a lot of noise. So we ended up using some filters, which I won't go into right now due to time, but we ended up focusing on just 32 top developmental genes and 11 top taxa, most of them being fungi. From that, I then filtered the correlations down to focus on the top 15 positive and top 15 negative correlations for each stage and care group. So I'll show you what the results look like. Okay, so here are the results. And as we can see, there's a lot happening here. There are a lot of patterns. So I'm only going to talk about a few that I found interesting. And again, this is just showing us the top 15 positive correlations here and the top 15 negative correlations for each of the developmental stages. The stages are colored here by the links, and so they're associated here between care and no care. And so the first takeaway from this is that in each category, there's typically one fungus in the center here, that appears to have the most top correlations, and it's often either just escosphera or Postdoctoral. Now, we might have expected Aspergillus to be the center in some of these categories, especially in no care, because its abundance was so high. But the important thing with this type of approach, or this type of an integrative analysis, is it really highlights that there are other important relationships occurring that might be missed if we only focused on abundance data. And so first, I'll just focus on those positive correlations, and I'll just talk about just some examples. So escosphera was central, meaning it had more correlations with many developmental genes in both care and no care groups. Well, we can see that it definitely had more strong correlations when mothers were present. And this suggests to me that the presence of mothers provides a less stressed environment, allowing for natural development to occur, even in the presence of a potentially pathogenic fungus. For example, escosphera correlated with the PIT gene, or pitchurine for I think it's called pitchurine, in callus. It's a gene involved with ribosomal biogenesis and protein synthesis. Both are very energy demanding processes. It also correlated with isocitrate dehydrogenase, IDH3G, in pupae. This is a gene involved with amino acid metabolism. And so these genes are energy demanding, and neither of them were found among the top correlations with escosphera for any of the other stages in this no care condition. And so what is escosphera? So it is also a problematic plant mold, which is known to cause chalkroot disease in honeybees. It infects them mostly in the larval and pupal stages, making them turn chalky white and mummifying them. And it's an infection that can spread easily. However, again, like Aspergillus, its effects are unclear or unknown in solitary bees, but we can still speculate that its presence might be inducing some stress in both care and no care treatments, but it would more likely be having a greater impact in the no care treatments, simply because the offspring are already deprived of mothers, and they're facing, and they don't have that protection from the mothers during early development. For instance, escosphera in no care correlated with more genes during the late larval stages, so the blue lines here, and the pupal stages, the pink lines here. Again, these are the two stages that it's known to have the most impact, at least in honeybees. And then escosphera positively correlated with the cellular respiratory gene, UQCRFS1, in pupal stages in the care group. But a lack of mothers resulted in the same correlation during the late larval stage here. This gene is involved with energy production, and energy metabolism is a process that is known to often increase under stressed conditions. So this result shows us that this might be occurring earlier in developmental stages when mothers are absent. And there was also another study that found that honeybee larvae infected with escosphera did show signs of elevated energy metabolism. So now looking at the negative correlations, Postdoctoral had many negative correlations with developmental genes in the care condition, whereas for the no care condition, it was escosphera again in the center. So Postdoctoral, it is a soil fungus, or it's also associated with decaying plant matter. And while it's typically referred to as a plant pathogen, it does produce secondary metabolites that are known to be toxic to insects. So here I speculate that its presence here is inducing some immunity response, even in brood reared with mothers. For instance, it correlated with the neurodevelopmental gene DSCAM2 during late larval development. This is a gene that plays a role in immunity as well, suggesting that healthy developing larvae, due to the presence of mothers, may be able to inhibit the effects of harmful fungi via the upregulation of immunity genes. But if we look at the no care group, Postdoctoral negatively correlated with DSCAM2, again in late larvae. So the blue line is shown here as well. And in fact, it correlates with several neurodevelopmental genes in both care and no care conditions in early and late larval stages. And so to me, this means that regardless of maternal care, Postdoctoral is affecting neurodevelopment and may cause nervous system dysfunction. And in fact, nervous nervous system dysfunction was found to occur in ghost moth larvae that were infected with Placellomyces. And overall here, we do see that co-expression patterns between gene expression and metatranscriptomic data across development have some indication of dysregulation of gene expression and microbial abundance, especially in the no-care groups. But the patterns here are quite complex and likely showing us a snippet of what would be the whole regulatory system and interplay of multiple genes and taxa. And so just to wrap up, this project is still ongoing, but here are some of the conclusions thus far. So our developmental transcriptome revealed that gene expression data is clearly influenced by developmental stage of Ceratina calcarata. However, maternal care did have some impact on gene expression, especially during the early larval developmental phase. Likewise, the absence of mothers also makes early larvae more susceptible to fungal and bacterial exposure, especially concerning its exposure to harmful fungi like Aspergillus. While Aspergillus was the main one we found, there were other plant and soil fungi that were elevated in no-care early larvae. I just did not mention them. And these can also indirectly be harmful to larvae because these other types of fungi are also known to cause pollen spoilage. So it is clear that mothers play an important role in keeping nests clear of fungal and bacterial outbreaks during early larval development. Finally, other top fungi such as escosphera and Placellomyces showed interesting patterns and top correlations with developmental and immunity genes across the stages. Again, this was surprising because we were expecting a lot of relationships to come up with Aspergillus because of its abundance and presence, but integrative analysis just shows us that there are these other relationships that we need to be watchful for. And then finally, our next step in this project is to also include a component of transcription factor enrichment and identify any TFs among our DEGs. So cellular responses to environmental changes are mostly handled by TFs, transcription factors. So specifically, we'll be looking into enriched motifs or binding sites for TFs, and then among those abundant motifs, what type of TFs are they most likely binding? In other words, what is the motif enriched for, and whether it be TFs that are involved with immune response, stress response, or development. And this will give us a more complete picture of how maternal care is influencing gene expression and gene regulation across development. And then finally, this project shows how combining transcriptomics and metatranscriptomics can be useful in assessing environmental stressors and their impact on bee gene expression and their microbiomes through development, and we can do this simultaneously, which is cool. And with that, I conclude my talk. Thank you for everyone, the Rehan Lab, for their support and their efforts with the project. Thank you to my supervisor, Sandra Rehan, and co-authors on the projects, and additional lab members for all their help and all the resources that I've been able to use at York University. That I end my talk. Thank you.
Perfect. Thank you very much, Catherine. There's so much information. It's so interesting to see
So
how
I can't hear anything.
little things
My
like
headset
fungal
is off.
interactions can interfere with, the immune response and genetics and all the rest. And I apologize for people joining in late. I have a really low, crappy voice with a cold, so please bear with me. Let's turn it over to the audience now. Does anyone have any questions for Catherine? Feel free to just mute yourself and ask questions, or you can raise your hand, virtually if your camera is on, or through the raise hand feature in Zoom. Not seeing any, so I'll wrap things up. It's just 1 o'clock now, or sorry, 12 o'clock now. So thank you everyone for joining in today, and thanks Kate for again a really amazing presentation. Join us next month in 2023 for next speaker, which just did change his schedule as compared to what was previously advertised. The speaker who was scheduled for January isn't able to present, but fortunately Trevor Sless is able to join us and present in that time slot. So again, that January 13th, Friday at 11 a.m. Eastern, join us. The same Zoom link will apply. And then also, if you want to save the date, we just decided yesterday we're going to have a social in February, kind of the dark months of that winter time. So February 9th on Thursday from 2 to 4 p.m., probably somewhere in Lumbers. It's kind of informal. Again, associates get together, bring some snacks in. We'll just sort of hang out, catch up with each other, and just have some fun get together in some of the dark days of winter. So with that, thank you again for coming. If you're celebrating the holidays, have a happy time. If you're not, hope you have a chance to sort of rest up and get ready for an exciting year of research in 2023. So yeah, that's it, everybody. Thank you for joining us.
Thank you.
And we'll see you
Thanks,
next
everyone.
one.